risk classification of HPV genotype was defined, and the cervical cancer screening handbook was formulated. His field of expertise is DNA and RNA viruses, human papillomaviruses, tumor viruses, cell cycle regulation, epidemiology. Today's lecture titles are Vaccines as Efficient as Strategies to Combat Viral Diseases in the Area of COVID-19. So I would like to hand over to Professor Ifner. Kindly start your lecture.
Okay, thank you very much. I would like to say hello to everybody. I know it's 2 o'clock p.m. in Pakistan. It's 10 o'clock in the morning in Germany, and I would like to thank the organizers for inviting me for this very nice talk. And I also would like to give my special thanks to Sumbala Sheikh, who helped me and assisted me in making this today possible. So I will turn now to my presentation. Okay, I hope you can see that. Is everybody able to see that?
Yes, we can see that.
Okay, great. Thanks a lot. Okay, so great, thank you. So this is a quick overview of the topics I would like to show you, and I would like to start a little bit with history. I mean, just to tell you about the basics about the vaccine, how it has been actually developed, what kind of immunizations we have using a vaccine, active and passive immunization. There are different kind of vaccines available. Usually you have adjuvants in the vaccine as ingredients that stimulate, in addition, the immune system. And then I will show you some data on how vaccines are generally developed. And then I would like to switch to the COVID-19 vaccines, and I will give you a short background on coronaviruses, which vaccines are in the pipeline now, what kind of vaccines are developed, some questions about vaccine dosage, and some common questions. Feel free to interrupt me anytime in this talk and just ask a question, raise your hand, or speak up. Okay, this slide gives us a history of the vaccines, and the whole thing started really a long time ago, and it became more known in the 15th century, where smallpox caused big pandemics in China and India. And there it was kind of a common knowledge that if you took some scabs from the pox that were caused by smallpox and grinded material into your skin, or blow it into the nostril, that you have that you can get some kind of protection. That was also then followed up to the 17th century. However, 20 to 30% of those who variolated themselves actually died of this procedure. However, that was a smaller percentage of those 50 to 80% that usually died after contracting smallpox naturally. So this is not a good way to do it. Now, in the 17th century, it was common knowledge among farm people that took the milk from the cows, that cows can also be infected by cowpox, and this disease can be also transmitted to the hens of those who milked the cows. And it was observed that people who got these lesions from the cow later on in smallpox outbreaks were actually protected. And Edward Jenner, finally, 1796, tested this hypothesis that cowpox infection could protect persons from smallpox infection by infecting an 8-year-old boy with cowpox, and he recovered fully. And later on, the same boy who was healthy was inoculated with fresh human smallpox. Sorry, a P is missing here. And it was found that the boy, in fact, is protected. Later on, in 1879, the first laboratory vaccine was developed by Louis Pasteur in Paris. Just by accident, he found that he could attenuate bacteria by oxygen, and later on, he developed the first vaccine against rabies in rabbits. So in the past, to summarize, we had kind of an inoculation of unweakened live pathogens, often done arm to arm or scab to arm, and often the patient became infected with smallpox, so this is not a good way to do it. The first vaccination was, as I showed you, done by Edward Jenner using cowpox, and because the cow in Latin is called vacca, the name vaccine is derived from this first experiment. Nowadays, vaccination are in that way that the vaccines contain microorganisms or virus in a weakened, live or killed state, or only viral proteins, so only some subunits of the whole virus. And they are used to stimulate the adaptive immunity to protect against infectious diseases, and this induces immunity. After vaccination, you can actually develop mild symptoms, but you should not develop the disease. So the aim of the vaccination is actually first to prevent the disease, but also to prevent the whole population from infection from others by achieving herd immunity, meaning if you are in the range of 70% of people of the whole populations that are immunized, then the infectious agent causing the disease is no longer able to spread in this population. And we have examples that we have the worldwide eradication of smallpox actually, and also the elimination of diseases such as polio from much of the world. Now, there are two types of immunization. Passive immunization, let's start on the right side. You have a natural immunity of the newborns because they acquire passive immunity from their mothers by antibodies that cross the placenta, and also antibodies that are present in the milk, and they are transmitted via breastfeeding to the newborn. And this gives the newborn protection for a few months, after which the newborn can also develop its own immune system. And nowadays, you can also use this by creating a kind of an artificial immunity. You can administer antibodies to non-immune individuals, and they are derived either from pooled or purified blood products, or they are constructed as monoclonal antibodies, and they can protect people after exposure to pathogens. The active immunization is quite different from that. You expose the immune system to an infectious agent, which is weakened, or only to subunits of this agent. And this triggers the immune system to make a B-cell response and to produce antibodies, and also to have a specific T-cell response. And if you have now, and this triggers, for example, here, the development of neutralizing antibodies. It takes a while, it takes up to 10 days or 14 days. And then you have the so-called memory B cells and memory T cells. And if the same infectious agent again comes in contact with your immune system, those memory B and T cells cause a much higher and much faster immune response, and you can actually combat the infectious agent. And this active immunization causes a long-lasting and sometimes lifelong vaccine-induced immunity. Now, what are the pros and cons about passive immunization? Well, it's difficult and costly to produce because you have to get these blood products, or you have to make monoclonal antibodies. You have to give it via intravenous injection, and you don't get any immunity. It only gives you a short-lived immunity because you have no immune response. However, it's quick acting within hours and days. It's much faster than a vaccine, and it can override a deficient immune system. So this is an option if you have no other ones. Now coming to the live attenuated vaccines. So there's an example like poliovirus. It's not used anymore nowadays to use a live attenuated poliovirus because it has only a few mutations in the vaccine strain, and these mutations can actually revert in the host infected with the vaccine virus back to the wild type, and then this wild type can cause another outbreak of polio. And this has been observed in the Dominican Republic, for example, and nowadays only an not live vaccine of poliovirus is used anymore. And you need actually not only a single dose, you need usually 2 to 3 dose regimens. However, if you use a live attenuated vaccine, the virus replicates in the cells in your body, and this produces the same complex immunological response like a natural infection. You have humoral response, so producing antibodies. You have a cell-mediated response producing T helper and cytotoxic T cells to a variety of antigens of this live attenuated vaccine. Now, the other way of active immunization is using inactivated viruses or purified antigen vaccines. So the immune response is then only to the components that are present in the vaccine and not to the full virus, which causes a lower cytotoxic T cell stimulation by cross-presentation. You get, however, satisfactory long-time antibody levels if you give 2 or 3 doses. And the antibodies, they can wane over time, but usually you have a protection for at least 10 years. The pros are the antigens, which are usually proteins and glycoproteins, make a good humoral immunity, but however a weaker T cell response. Now, what's in a vaccine usually? Now, the main ingredient in a vaccine, and this is a vaccine here, is water. In addition, you have preservatives and stabilizers. Then you have residual traces from the production process. So for example, if you look at the influenza vaccine that is nowadays still made in eggs from chicken, you have residual traces from proteins from these eggs. Then you have the active ingredient, which is a harmless form of the virus or a subunit, as I already explained to you. And then in some vaccines, you have also adjuvants like aluminum, for example, that just cause a stimulation of dendritic cells and activation of complements, and also release of chemokines. So it makes actually, it acts as a stimulator of the immune system in addition to the antigen you provide. Now, vaccine development. You probably have heard you have phase 1, phase 2, phase 3, and this is shown here. In phase 1, usually you have a small population of 20 to 100 healthy volunteers, and you want to answer the question if the vaccine is safe. Are there any serious side effects? How does a vaccine dose relate to any side effects? So you try to find the right dose in the phase 1. And also you want to answer the question if the vaccine at all causes an immune response. So now, if this is satisfactorily answered, then you may enter phase 2. And all this is highly regulated. You have to get always, you have to show the data to the official authorities. And then only if they are satisfied, you can go to phase 2, where you usually have several hundred of volunteers. And this time you want to answer the question, what are the most common short-term side effects? What's the body's immune response? And now the first time there's a question, do you see any protective effect of this vaccine? And if all these questions are satisfactorily answered, you may enter into phase 3 after admission by the authorities. And there usually you have large trials in the range of 30 to 40,000 people, and you make you split them in a fully blinded in a fully blinded controlled study into a group that actually gets placebo, and the other group, 50%, that gets the real vaccine. And this time you want to answer the questions, how does disease rate compare between people who get the vaccine and those who do not? So you need exposure of this group to, for example, the virus. And that's why at the moment, most of the studies for the corona vaccine take place in Brazil, because there we have a high rate of disease. And then how well can the vaccine protect people from disease? And if this is answered satisfactory, then you have a very complicated approval process where the authorities look at all the data, and only if they are 100% convinced, you get an approval. And then phase 4 starts after approval and after the vaccine is administered to normal people. So outside of these studies, then we have usually a 10 years phase where all vaccinated people are actually observed for a longer time. And please keep in mind the FDA and also the EMA, so the regulatory authorities of the USA and of the European communities only approve a vaccine if it's safe, effective, and if the benefits outweigh the risk. And then the researchers continue to collect data on the vaccine long-term benefits and very rare side effects that you cannot observe in a population of 30,000, but maybe in a population of a few million. As an example, I show you here the vaccine against human papillomaviruses, which is usually my field of expertise. And human papillomaviruses can cause, when they make a persistent infection that is lasting for at least 2 years, pre-cancer, and this pre-cancer can develop into cancer. Men and women can be infected, and women can develop cervical cancer, and this can take place or in women between 35 to 59, and in men, these viruses can mainly cause tumors in the oropharynx, so in the mouth and throat, but also in the genital and anal areas. And now there's a vaccine available to prevent these precancerous cancerous stages of cervical cancer up to 94%, and also anal lesions about 76%. And how does this work? If you are not vaccinated and you get infected and you develop a persistent infection with human papillomaviruses, you can get tissue changes in the cervix, for example. And if those tissue changes are not detected, they can develop into precancer. And it takes another 7 to 10 years if these precancers are not detected, then you may develop a full-blown cancer. If you are vaccinated, you prevent the persistent infection, and because you take out the major risk factor, which is a persistent infection, you get no precancer and no cancer. And the recommendation of the vaccine is for boys and girls in the age from 9 to 14, and also they get 2 doses, and from 15 to 17, they get 3 doses. Already in 2018, worldwide, 270 million doses have been given, and there were no serious adverse effects. Now, what is this vaccine built of? This is a picture of a papillomavirus. I'm not going to go into detail in that. What you see here is the outer shell. And this outer shell, when you look at the genome of the virus, it has 2 proteins that build up the capsid. So the capsid is built out, the outer shell is built out of these capsid proteins, and the major capsid protein is L1. And now what they do, they use this outer shell, the Tetra Pak actually, but there is no viral DNA in this outer shell. So it's a virus-like particle that contains no DNA. And this virus-like particle is then given as a vaccine for our immune system. It looks like a natural virus. However, it does not. It's produced either in yeast or in insect cells. And this L1 protein has the ability to form pentamers, and these pentamers can aggregate directly into a virus-like particle. And as I said, this empty particle contains no viral cancer genes, is immunogenic and very safe, and has been, as I said, given to a few 100 million people already. And in Australia, where they started very early, we see a dramatic decline of those HPV types that are in the vaccine. So how does it work? The vaccine then makes antibodies and a T-cell response, and these antibodies can actually prevent the virus from entering cells, so you get no infection. If there are a few cells infected, the T-cell response will kill those infected cells. And this is how it works. Nowadays, we have a vaccine on the market that has 9 types, 7 types against cervical, 7 types that are known to cause 92% of cervical cancer, and then 2 types that cause genital warts. And in Australia, in the vaccinated groups, there is genital warts are no longer found. Now let's shift to coronaviruses. How do they look like? They are now different from HPV because HPV is a DNA virus. Coronaviruses are positive single-stranded RNA viruses with a large genome up to 32 kilobases. These are the largest RNA viruses on the planet that affect humans. And you see here a picture of them. And this is a cross section. Inside, you can see the viral RNA that is also associated with nucleoprotein. On the outer side, you can see those projections. And these projections are very important because they are actually the molecules that dock onto cellular receptors. And so they make the connection to the plasma membrane of the cell. And they are also the target of antibodies that can neutralize this virus. And they are also used in the vaccine, as we will see later on. So these large particles are also called spikes, the spike protein. And coronaviruses are not new to us. We know that since 1970 that respiratory infections, harmless respiratory infections, can be caused by certain types of coronaviruses. They make a common cold, and this is nothing. So OC43 or NL63 or NL229 are those viruses. But we have seen, starting in 2003, there can be also other forms, like the first form of SARS that caused an outbreak mainly in China. And we had a mortality rate of 10%. And later on MERS, which was mostly found in the Arabian states. Now, our SARS coronavirus 2, which is now causing this pandemic, belongs to the beta coronaviruses. And as you can see, this is a kind of a phylogenetic tree. And these harmless viruses that cause a common cold are also closely related to this SARS coronavirus 2. If you go, on this slide, you can see the spike protein is able to bind to a receptor for the angiotensin converting enzyme. The angiotensin protein is a protein that regulates blood pressure by vasoconstriction. And this receptor for this molecule is used by the spike protein of SARS coronavirus. First, it needs to be cleaved, and then if all these things happened, the virus can enter the cell. We will talk about this in the next slide. This slide shows you the genome of SARS-CoV-2. It's about between 29,000 to 31,000 nucleotides. You see the largest genetic reading frames encode the replication polymerase complex. And this is important, and also the proteases. And this is important because this is an RNA virus that has a proofreading activity in the polymerase. So it can actually detect if wrong nucleotides have been built in the new developing strand from an RNA genome to the next RNA genome, and they can be removed. This is normally not the case. For example, you cannot find this in influenza or in HIV, and this is why those viruses cannot grow much bigger, because, for example, HIV causes at least every 10,000 nucleotide has a mutation. So no genome that is made from the original genome has not a mutation. in the case of HIV, and the mutation rate in SARS-CoV-2 is much lower, and this allows this virus to grow, to have such a large genome. However, it makes recombinations. We will come to that later. Now, this plus strand RNA can be directly translated into a large viral protein, and this protein then later on needs to be cleaved by proteases into the single viral proteins that you can see here, like the spike protein or the envelope and membrane protein. There is a lot of speculation where it comes from. It has been found that it shows the highest sequence homology to the horseshoe bat virus in China. However, the receptor binding domain of the spike protein here is not very similar to the one of the horseshoe bat virus, but more closely related to a coronavirus from pangolins. And pangolins, there is a large travel from pangolins from Malaysia to China, where they make Chinese medicine out of it. And so it's possible that actually are two different events of transmission from pangolins to humans and also from bats to humans has allowed to develop this SARS-CoV-2 virus, which has a high affinity to the human ACE2 receptor. Now, let's come to the disease. Usually, in a large percentage, we have asymptomatic disease, about 42%. The incubation period is in the mean about 5 days, but it can range from 1 to 14 days. And about 80% of the people have a mild disease with fever, fatigue, dry cough. You can find in the CT, ground glass opacity changes, and you can also develop pneumonia, and this can lead to severe diseases that can, at the end of the normal duration of the disease, which is about 14 days, that can develop. And this is also age dependent. So children below 10 years are usually rarely infected and do not spread the virus infection as good as adults. If you are below 50 years, usually you will not get the severe disease. The biggest risk factor for getting severe disease and critical disease like acute respiratory distress syndrome, multi-organic failure, acute cardiac injury is male, gender, and high age above 68 years. Now, this gives you an idea about how it looks like. Let's go here first. So this is a CT image showing the lung of a 33-year-old woman with a mild disease. However, you still can see this plaques here in the lung, which is a fibrotic development in the lung. However, that can revert. But if you look here at these, at the same, at a CT picture from an 83-year-old woman that has a very severe disease, you can see a bronchial thickening, consolidation of the left lobe and interlobular septa, and this woman is no longer able to breathe by herself, so she has to become incubated and get And when you look at the viral load in the upper respiratory tract, you have a higher viral load than in the lower respiratory tract, where you have the more severe disease. That means it can happen that in severe diseased people, you will find no virus any longer at the end in the upper respiratory tract, but only in tracheal secretes. Again, one major factor driving infectivity and spread is how good does the spike protein of SARS-CoV-2 bind to the cellular ACE2 receptor. How good is it cleaved? If you have a high affinity and a high binding capacity, you need less virus for infection and the transmissibility is increased. That's the whole replication cycle of the virus. And you can see again, after binding to the receptor, it can either be uptaken in an endosome from which is then released by the drop in pH, or it can directly fuse on the cell membrane, and the RNA is then, as I said, translated by the cellular replication machinery into a large polyprotein that needs to be cleaved by proteases into the single viral proteins. Then we need an enzyme that is not present in a mammalian cell, an RNA-dependent polymerase. And this makes from the plus strand RNA a minus strand RNA, which is then used again as a template to produce again the new viral genomes, which is again plus stranded DNA. And this is then packaged into a membrane vesicle, and this membrane vesicle is then transported through the Golgi apparatus to the surface of the cell and is released. So generally, you have a higher viral load in the nose than in the pharynx. There is some reports about detection in blood samples. It has not been found in urine. It has been also found in feces. However, it's not clear if this contributes to the infection, to the spread of the infection. Interestingly, also people who are asymptomatically infected are highly infectious and can transmit the virus as good as symptomatic infected people. And the worst thing about the virus is usually it can somebody who is infected and has not yet developed any symptoms, so is still in the incubation period, is highly infectious and contagious. So it can spread easily the virus without being even notifying that that the person is ill. This I already said. So we have an unusual high virus titer, actually up in the tracheal, up in the oropharynx, up to 10 copies per milliliter. We have also some cases now up to 10 to 9 copies per milliliter, and in the swab, in the feces, there have been no infectious viruses found. I said already about 43% of the laboratory confirmed cases were asymptomatic. And if you look here, what happens between day 0 and day 14 after the symptom onset, usually after about day 5 or 6, you develop antibodies against the virus. And if you try to grow the virus from swabs of the pharynx in tissue culture, for this you need a lab with a biosafety level 3, which we have here. You can actually get cultures up to day 8, but then the viral load drops and you are no longer able to isolate the virus from these samples. Now, this is about the outbreak, which started about actually probably around November already, with unnotified events in China. And then there was a cluster of 27 pneumonia cases in Wuhan. And it was found that there's a spread at the wholesale human seafood market. And this has been closed. And after China realized that there is an outbreak, you may remember they closed down large cities and nobody was allowed to leave any longer or to get in. And within weeks, the pandemic spread from China to other parts of Asia, to Australia, to North America. And then you can see, even at the end of February, large parts of the world were infected with this strain that has been initiated from Wuhan in China. Nowadays, we know that this Wuhan lineage or strain is no longer circulating in the population. However, we have the development of new variants, and I only show you 3 of them, which are quite famous at the moment. And there has been in September 2020, the first cases of a new strain called B117 in Southeast England. And within weeks, it was the prominent strain in these parts. And nowadays, it's the driver of the pandemic in the United Kingdom. Another development was in South Africa, where the strain B1351 has been found. Both of those strains seem to transmit much better, and this is due to mutations in the receptor binding domain of the spike protein, which allows to have the spike protein a higher affinity to the receptor on the cell, and therefore you need less virus, and therefore you see actually about a 50% increased infection rate of those strains. Another strain that popped up in Manaus in Brazil and was transmitted to Japan also has other mutations, not in the receptor binding domain, but especially in those regions that are recognized by neutralizing antibodies that can actually prevent the virus to enter the cell. And this is a more concerning thing because it might affect the efficacy of treatments with monoclonal antibodies or the efficacy of vaccines. I will come back to that. Now, what vaccines are now have been produced or are in the process of production against SARS-CoV-2? We have DNA-based vaccines where you actually inject DNA of viral genes into the muscle, and the cells take up the DNA plasmid and make proteins. And this has been done by Sanofi with not such a big success, I have to say. And with Inovio, we have viral vector vaccines where they use, like in the AstraZeneca vaccine, they use a chimpanzee adenovirus. And on this adenovirus, they put on the surface the spike protein from SARS-CoV-2. And this vaccine has not been authorized, but is very close to the authorization. Merck has given up in the meantime because they had no good immune response, and other candidates are still in the game. The very most interesting vaccines, because they are completely new, are the RNA vaccines that are used by BioNTech, Pfizer, and by Moderna. Both of those vaccines have been authorized in the moment. And what they use, they only use a messenger RNA coding for the spike protein. And this seems to be a quite successful approach, because this will probably revolutionize the whole field of vaccination, because they are quite quick they can quite quickly be produced, and they seem to be very effective. We have efficacies in the range of 94 to 95%. And there's another company in Germany, CureVac. So BioNTech is a company from Germany together, and they made this together with Pfizer. And CureVac is a company in Germany, in Tübingen actually, where I'm from. And they are close to become authorized. Then there are subunit vaccines where they just take viral proteins, usually the spike protein, but also some other proteins. Some companies use inactivated vaccines, so they use the whole virus and inactivate it, which is quite easy, because with heat or with formaldehyde, you can kill the virus, because if the virus has no lipid envelope anymore, it's not infectious, but you still have all components of the virus in the vaccine, which is, however, no longer infectious. Nobody has tried to develop a live attenuated vaccine of SARS-CoV-2. There has not been enough time. And I have to say, all these vaccines have been produced in an incredible fast way. And we have seen the fastest process of vaccine development ever in this pandemic area. Now, again, we have two messenger RNA vaccines, Pfizer BioNTech, that both encode for the SARS-CoV-2 spike protein. Moderna is the other company. They have been authorized in the EU and the US, both of them, and one also in Israel and in Canada. Then the AstraZeneca that has been developed in Oxford is a low-cost vaccine, specially sought to fulfill the needs of the low-income or medium-income countries, because it will be sold for $2 to $3 per dose. And this is, as I said, this is a viral vector vaccine using chimpanzee adenovirus vector expressing the SARS-CoV-2 spike protein. And they have been already approved in Argentina, India, and there was an emergency use approval in the UK. And all these three vaccines are currently given to people. Now, there are two whole inactivated vaccines by a company in Wuhan, China, and by another company, Sinovac, where we don't know who funds this. And there is this famous Sputnik vaccine from Russia, which is a viral vector where also using adenoviruses expressing the spike protein. Now, just to give an example how these studies looked like, the Pfizer-BioNTech, they enrolled about 44,000 people at 150 clinical sites in 39 US states, and they tried to have the ethnical distribution of their population. They included also in 45% people who are between 56 to 85 years old, and this vaccine has been approved and is given at the moment. Moderna, they enrolled 30,000 people at 89 clinical sites in 32 states. Again, the racial ethnical distribution was, they tried to make a representative picture of the population. It was also given in 64% to people 45 years and older, 25 people also 65 and older. And AstraZeneca enrolled 30,000 people at 117 clinical sites. And CureVac is at the moment enrolling 36,000 people in Europe and also in South America. And they are in phase 3, and and they hope to finish this phase 3 in March, April. Now, how does the vaccine work? So the RNA vaccine is actually packaged in liposomes, and this mixture of liposomes with the RNA is given, is injected into the muscle of the arm. And the Oxford vaccine is this adenovirus vaccine with the spike protein of SARS-CoV-2 and is also injected into the arm. And when the vaccine enters cells, it starts to produce a spike protein, and then the body reacts with the immune system reacts, making antibodies and also T cells specific for the spike protein. And later on, if this patient who, not the patient, if the person who was vaccinated catches coronavirus SARS-CoV-2, the antibodies and T cells are already there, and they are triggered, and they can combat efficiently the virus. Now, what is in these vaccines? Mostly RNA in the Pfizer and Moderna vaccine. Then we have a combination of different lipids, as you can see here, some buffers, but no adjuvants. And the AstraZeneca contains this weakened adenovirus with a spike protein, also some other components, sugar and magnesium chloride hexahydrate as some kind of an adjuvant. But it has to be absolutely clear, none of those vaccines can cause COVID-19. They only can give you a protection against COVID-19. How effective are the vaccines? Now, there are two terms you should know. Efficacy means a vaccine protection that is measured in these randomized controlled trials under optimal conditions. And the vaccine effectiveness is later on observed in the phase 4 trials. So after the vaccine is given to normal people. Now we have only efficacy data at the moment, and you can see Pfizer-BioNTech has an efficacy of 95%, Moderna 94.1%, and AstraZeneca had a little bit of a hard time because when they had to change actually the regimen in the phase 3, because when they gave 2 full doses as they wanted with 10 to the 8 viral particles they had only an efficacy of 60%. But when they switched to give half dose first and then a full dose second, then they achieved also 90% efficacy. Again, how safe are the vaccines? All these vaccines have to be found safe, well tolerated. They can cause temporary side effects as fever, headache, a sore arm. And this is a good sign because it shows you the vaccine works. Your immune system is responding to a new antigen and is producing antibodies and T cells. Most of the vaccines are given in 2 doses, 3 to 4 weeks apart. It takes about 1 to 2 weeks after the second dose to have a full protection. After the first dose, you have only about 50%, and therefore it's absolutely required to have a second dose to have a full protection. We don't know at the moment how long these vaccines will be protective. Especially, we don't know how good they will protect against new variants that are at the moment developing. There are many more than I have showed to you. And this is something we will have to find out in the future. It may be that we need to have regular shots against SARS-CoV-2 to prevent the disease COVID-19 on a regular basis, like the flu shot, because it's also a seasonal disease, which is popping up in the winter months. A lot of people ask, will I still need to wear a mask? Yes, you do, even if you are vaccinated, because a large number of the people in the community need to get vaccinated before we have a herd immunity and transmission stops. So at the moment, as long as we do not have herd immunity, you still have to use the vaccines, the masks, sorry, especially because it's a little bit like in the case of the HPV vaccine or other vaccines. You cannot fully prevent a transient infection, and that can allow also that some of the vaccinated people also will spread to a very small amount virus. We don't know if this can cause infections, but as long as we don't know more, wearing a mask is absolutely necessary. It is safe, even if you already had COVID-19, to get a vaccine, and you should actually get vaccinated because you get a longer protection against the disease, because we see a waning immunity in some people, especially if they had only mild symptoms. The antibodies will last only a few months. And even if you have a positive antibody test, it doesn't mean that you cannot be reinfected, and still you should get the vaccine. And with this, I would like to thank you all for listening, and I'm happy to take any questions.
Thank you very much, sir, for such an interesting talk. Now our forum is open for questions.
Dear friend, Professor Thomas Hifner, this is Iqbal Chaudhry.
Oh,
and
my
what a
very
wonderful lecture
greetings
as
to you.
always. Thank you. I just wanted to know, I mean, there are lots of genomic surveillance, people are trying to understand how this virus is mutating. And, with my basic understanding of biology, this virus perhaps would mutate much faster in vaccinated people because it's a simple logic of survival of the fittest. So how do you see this thing happening?
This is why I actually said you need to make sure that you get a second dose, because the worst thing would be if you have only a small kind of protection, maximum 50%, and then you get infected, because we have these people that get infected shortly before vaccination or afterwards. And this is dangerous because, as you said, you have a selective pressure already that is ongoing, but you have not really good protection. and then you can develop new strains that escape immunity. It has even been shown in publications where they simulated things like that, where they infected cells and then they put sera from convalescent patients on these cells, and then they observed also the development of new vaccines, of new variants, sorry. So from the data we have seen so far from the vaccine trials, it seems to be very exceptional that somebody can reinfect it with the same variant that was in the vaccine. But luckily, we are now in the status of having these messenger RNA vaccine platforms. So within about 6 to 8 weeks, they can react and they can produce a new vaccine. So it doesn't take half a year like with influenza to get a new vaccine. And so I think we need a good surveillance, and we are just trying to build up this in Germany. We have somehow, our politicians have not really reacted to our questions and to our demands to set up a genomic surveillance here in Germany. Now we are starting to doing it much too late because UK and Denmark is since half a year doing this. So we, in combination with having a good surveillance where we sequence all the genomes from positive patients and the messenger RNA vaccine platforms that can actually react to new variants, I think we are in a quite good shape.
Thank you. Thank you, sir. Any more questions?
I would like to ask a question.
Yes, sir.
Am I audible?
Yes.
I'm Ariva from Healthcare Biotechnology, NAST Islamabad. I would like to ask that you mentioned that we would be needing the regular intervals of COVID-19 vaccine, just like flu shots. So my question is that we know that flu shots are given in the season of autumn and winter. So what would be the season of taking the booster shots of COVID-19? Like, would it be winter or summer? What would be the predictable season for booster shots of COVID-19?
Exactly the same as the flu shots, because it's quite well known that humidity, so relative humidity in the air, is affecting a lot the stability of the virus. So if we have a low humidity, the virus is longer stable in the environment than as if you would have a high humidity. Even 12% can here make a dramatic change. And it's also known that outside, when you have a sunny day, there's a lot of UVC outside, and UVC is a very big killer for this virus. So we have also shown this in the lab. Even if you use UVC on contaminated surfaces with SARS-CoV-2, within seconds you can completely inactivate the virus. And we are using this technique also in other techniques like plasma filters to develop devices for inactivating this virus.
Thank you. I have one more question, if I'm allowed to ask.
You're welcome.
I want to know that you mentioned that none of the vaccine manufacturing companies worked on live attenuated virus. So what are the obstacles behind them?
Well, it takes a while. I mean, usually you get attenuated viruses by continuously passaging the virus in tissue culture, usually in cells where it does not really very well replicate. And then you have to wait till you find these attenuated forms, and there has not been enough time. And at the moment, I don't think you will get any authorization for a trial, especially when there are such good vaccines already available.
Okay, thank you, sir, for sharing such good information with us.
You're welcome.
Hello, Dr. Refnet. This is Dr. Rashid from National Institute of Virology.
Nice to hear from you.
Thank you so much for a very informative talk. So my question is, as you mentioned, that the patient with this complicated infections, there are chances that the virus load is more in the lungs, in the lower respiratory tract, and perhaps the upper respiratory tract has very low or no virus load. And we have a similar observation in our lab also. My question is that there are some patients who got infected. They are RT-PCR positive in their first week, and then they gone through the infection. They got symptoms, and then they recover. But after many weeks, they still have RT-PCR positive. How do you see it?
We have the same observation. I mean, it's common if you have immunosuppressed patients, like patients that have received a stem cell transplantation or so. So we have here, for example, patients that are now since 10 weeks in intensive care, and they still have a CT value of 25 in the pharynx, but nothing in the tracheal secrete. So it's the opposite, actually. So a special threat is actually coming from people that are immune compromised, because they also give the opportunity to the virus to mutate and to make new variants. It is also observed in patients that are not at the intensive care, and we have no real good answer at the moment. While we have some shedders that shed up to 10 or 12 weeks, we have also seen cases of reinfection of people that really had their first episode in March. Then they became completely negative in multiple sequential PCR tests. They developed antibodies, and they got reinfected. And it was not even another variant. So that's what I said, because in the first period, this patient had only mild symptoms. So probably the immunity waned over time, and the antibodies were no longer there. Yeah, but to answer your question, or to say it's not clear at the moment what causes these long time shedders, actually.
Okay, thank you so much, sir.
There is an online question.
Yeah, I have one question. This is Dr. Atya from ICCBS. I would like to ask that there are many observations that asymptomatic COVID-19, I would say patients, but they are not patients. They are completely healthy, and we have actually observed during our screening, because in National Institute of Virology, we have screened more than 100,000 uh, positive samples, and among them, we find that there are asymptomatic people, and so what would be the criteria that people need to be tested for the COVID-19?
Well, ideally it should be a mass screening, but that's not possible. So what we do here is we test everybody who is coming to the hospital and is taken up into the hospital before it is been taken up, just to prevent a spread here in the hospital. And this is a very good question, because usually you test symptomatic patients, but if they are not symptomatic, there is no need, and there is then you would actually need to test everybody, and that's not possible, because we are already at the edge with the capacity for testing, and also with all the material we need for just doing these tests. There is we are seeing a lot of problems to have all the chemicals. There's even a shortcut of liquid CO2 at the moment, so it's really it's difficult. Well, as I said, ideally everybody should be tested. That's not possible, so you have to restrict the testing to prevent outbreaks in hospitals, in nursery homes, and yeah. in other critical places.
Thank you.
Any more question?
There are two questions online. I can see. One is from Munir Ahmed. Mr. Munir Ahmed is asking, so far symptoms of vaccines remains in cases after doses given to them. Does the vaccine have any follow-up effects as we see in coronavirus onset in healthy individuals?
I'm not sure if I really understand the question.
Yeah, I think I also did not understand because it's probably some typing error or something like that, but
just
okay,
to
we
make
can
clear,
move forward.
No, just to make it clear, the vaccine will cause no COVID symptoms. It only causes unspecific symptoms as you stimulate the immune system. And this is actually good if you get fever, headache, fatigue, or whatever, because it just shows you the vaccine works. That's all I can say to that question. Sorry.
Sir, I have one question. My name is Dr. Samia. I'm a microbiologist. Sir, you gave a very informative talk. I have one confusion. There are a lot of high age group people suffering the COVID infection very seriously. This age group, the immune system is very low. How can manage to drive these antibodies? How can make? Because our literature shows that after 50, the antibodies develop mechanism is slow because the bone marrow unfunctioning. How can manage the vaccine to develop the antibodies?
Yeah, but this works also for influenza and for other vaccines, you know. So it doesn't mean that if you are old like me that you will not get any immune reaction or immune response after a vaccine. It may not last as long, and so you have to repeat it, but still it works. So you're right. In generally, the immune system is waning as the older you are, and this starts already at the age of 20, by the way. But nevertheless, vaccines have been shown to work, and also in elderly people.
How much booster dose planned to the vaccine, the one vaccine, these vaccine, how can develop the booster dose? Is it 1 month, 2 month, and 3 month?
No, it's usually after 3 or 4 weeks you get the second dose.
Life of these vaccine?
Sorry, I didn't hear.
The shelf life of these vaccine.
Oh, that is a big question, a big thing, yeah, because in the approval studies, the Pfizer-BioNTech vaccine, they used a vaccine that is stored at minus 70 degrees. And this is now the problem because they have to go along the approval guidelines. And so the transport chain has to be under these conditions. And then they are stable at room temperature for about a day or so. But Moderna is using minus 20, and it's the same vaccine actually. And I'm quite convinced that even under minus 20 degrees Celsius, also the Pfizer-BioNTech vaccine will be stable. However, they have been approved under these conditions. And that's a big problem because in low and middle income countries, you cannot have a transport chain under minus 70 degrees in every region of the country. Yeah, it's also in Germany a problem. You know, I have to say.
Okay, thank you so much. If we have no questions, thank you very much, sir, for your time and for this wonderful talk. Thank you.
My pleasure. I would like to thank you everybody here in the audience, and stay healthy, please.
Thank you, Dr. Ifner. We would like to stay with us for a few more minutes.
Okay.
Thank you very much. Thank you everyone for joining us. I'm hopeful this workshop was fruitful and memorable for everyone attending in person and virtually. With this, I would like to invite Dr. Sumbala Sheikh. Dr. Sumbala Sheikh has been very instrumental in
organizing this event along with Dr. Atiyatul Wahab. Dr. Sumbala, I invite you for your remarks.
Thank you so much, Hazima. Can everybody hear me?
Yes.
Okay, thank you. Respected guests, dear participants, and my worthy colleagues and honorable speakers, good afternoon. It's a pleasure and an honor for me to be here with all of you for the final session of this hybrid workshop on virology. Although we all know that we are not able to meet in person because of the COVID-19 pandemic, and it has limited us to only virtual contact most of the time. But I think that in spite of all its limitations, it is important we continue to debate over these issues. The lockdowns and restrictions, they have hindered our lives to such an extent, but still in the meantime, the pandemic also has helped to focus our minds on how to move forward. And I think we as human beings, as scientists, as social beings, we are determined to emerge from this time of relentless crisis with a brighter future. Because viruses do not stop at borders. And for this reason, coordinated international strategies to fight viral diseases is more effective than trying to overcome them alone. So I hope and wish we carry on such internationally coordinated events on a regular basis. In the end, I would like to thank Professor Iqbal Chaudhry, Coordinator General Comstech, Dr. Atiya, Dr. Rashid, Comstech staff, including Mr. Aftab Zedi, Mr. Usman Kudus, Mr. Naveed, Ms. Khazima, and their teams for taking care of the organization and coordination of this event, hospitality, and virtual delivery of this event. Special thanks to the IT teams at ICCPS for their continuous work. Last but not the least, I would like to thank our honorable resource persons, Professor Dr. Thomas Ifner, Professor Dr. D. Liu, Professor Dr. Khalid Said Khan, Professor Dr. Bertram Fleming, Professor Dr. Daniel Zauter, Professor Dr. Hong Ping Wei, Professor Dr. Wasim Jafri, Dr. Ji Kyun Woo, Dr. Mohammed Rashid, Dr. Sabah Farooq, Dr. Ishaq Khan, Professor Dr. Anders Balne, Dr. Almaz Javeen, Dr. Hana Iqbal for dedicating their valuable time in sharing their research and expertise during this virology workshop. Thank you all.
Okay, I'm sorry. Thank you, Dr. Sumbala. With this, I would request Dr. Ifner to please say a few words to our aspiring biologists and virologists who are present here today.
Thank you for giving me the word. First, again, I would like to thank again all the organizers and especially Professor Iqbal, where we have a kind of a long-standing relationship already, and Dr. Sumbala Sheikh also for their continuous help and support. And I think it is very important at the moment that we focus all our efforts to go on and fight this pandemic, not only by making new vaccines, but also by making therapeutics available to people who are deceased. First steps have been already seen by making monoclonal antibodies. Some of them, however, do not work anymore. Some of the variants, like the ones from Eli Lilly or from Regeneron, seem not to work as good. However, we still have to keep in mind, even if we see a drop in the efficacy of protection by neutralizing antibodies, it does not mean that the vaccine or the therapy will not work at all. We might see a kind of a drop, but we still have T cell responses and other things like that. So we all have to work together and fight this this disease and that will not be the last new virus that is showing up on this earth. And so we have to be constantly aware, set up a good surveillance system, yeah, and especially exchange our information with all people from all over the world. Thank you very much for participating, and I wish you all the best. I can't hear you.
Paul Chaudhary for his remarks.
Auzu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Good afternoon. Assalamu alaikum wa rahmatullahi wa barakatuh. My very dear friend, very valued speaker, Professor Dr. Thomas Ifna, very dear Sumbala Farooq, distinguished resource persons, delegates. This event was extremely important. I mean, one understand the relevance of such capacity building events. And if you see in the context of science, in this whole episode of unprecedented pandemic, the only thing which work successfully is science. When economies fail, when international cooperation miserably failed, when countries fail to support each other, uh, even relationship failed, the only thing which worked was science. Science has really worked very well. It has matched the pace of the spread of pandemic. Science fulfilled the promise for which we all have invested our emotion, our efforts, our money, and our resources. Science is certainly a success story. But where it actually all happened, we all need to understand it all happened in global north. It happened in countries like Germany, it happened in countries like UK, European countries, perhaps. So countries, take it. So countries in the global north have contributed. And as I said during the inauguration also, if there was no science, many of us were not here. Our existence was in real threat.
I can hear you perfectly.
Me
Dr.
too.
Can you hear us?
Yes,
Dr.
yes, I can hear you.
Yes,
Yes,
perfectly.
yes, I can hear you clearly.
Okay, so okay, so it is extremely important to develop institutional infrastructure and human capacity. Uh, and I recall this wonderful visit of Sumbala and Professor Ifna to Karachi well before COVID-19. It was in 2019 when we were discussing the establishment and capacity building of the National Institute of Virology. And even before that, I would like to remind all of you, and especially to Sumbala and Professor Ifna, our great founding father, Professor Dr. Wolfgang Folter. I went
Yeah.
to him all the way to Tübingen because we wanted to establish a virology institute. And he knew that we were in deep trouble. We didn't have many experts, we didn't have infrastructure, we didn't know how to establish a virology institute. And this great soul, this great man actually have taken me to Professor Ifner's laboratory. And we had the first discussion of the establishment of National Institute of Virology, which is now a reality. Uh, the genesis taken place. Well before COVID-19, there was a vision that pandemics and viral diseases are common and affecting people's life. Of course, we have never thought the magnitude of a problem which was forthcoming, but we knew that viral diseases are extremely important for us to work on. and there was no national facility for research on viral diseases. Now we have one. And of course, it need to be strengthened further. For that, we need to have cooperation and guidance from the people of Professor Ifner's stature. So we are absolutely fortunate to have great friends of ours who are willing to help us. I think COVID-19 has also given us an extremely important perspective. The perspective is that you can't really hide knowledge. You can't, in the disguise of biological weapon, you don't really refuse other countries to train and develop their capacity in the field like virology. You can't do that because the field of virology is extremely important because these diseases are not localized. They can't be one country problem. With the mobility we have, it is a global problem. Therefore, capacity need to be built locally while we have alliances at the global level. So, what I would like to urge you that from this wonderful workshop of ours, which was actually held in a blended format, you have heard some of the best minds, some of the finest virologists in the world have actually given you the prospect of recent development. And this is the reason why it is important for you to spread this knowledge further. I also urge you that somehow you develop a kind of a network of virologists in the country because we are very few and we are spread in many institutions, and that dilute the impact of whatever human capital we have in this important field. So my recommendation would be that form a network of virologists, and Comstech would be happy to support you. We'll be happy to link you with great experts in the world who are so well-meaning and who are willing to help you in your pursuit of excellence, and also to develop your own institutional linkages. For that, feel free to submit your project to us. Visit Comstech website. You will find lots of opportunities for you to travel to different places and to get necessary funding for at least small laboratories. This I wanted to mention you in the context of expectation. Science has performed very well, and therefore people have lots of trust on science. But at the same time, there is a downside is that they expect you to do that same thing, what happened in the West, without understanding that you work in a very constrained environment. So my recommendation to you is that excel, develop your capacity, form network, make this critical mass of people working in the field of virology through a networking, and then use whatever resources we have available within the country to carry out good quality virology work. Our responsibility, both at Comstech and also at ICCBS, will be to link you with the good institution abroad. So I'm absolutely delighted that both of my team in ICCBS and Comstech have done wonderfully well in organizing this excellent event. I am nothing else but a gratitude to all of you for your presence here, for your active participation. And it's not the last event in this field. We would have series of events in this field, and we will continue to benefit from the spirit of Professor Folter's, great mission of strengthening science in Pakistan. So this workshop is dedicated to one of the most wonderful persons we ever had, a great friend of Pakistan, Professor Wolfgang Folter. Thank you very much.
Thank you.
So congratulations, Professor Iqbal, for the setting up of the National Virology Lab.
Thank you, sir. I would like to thank all our participants and resource persons for their time. And with this, we come to end of this event. The tea is served in the adjacent room. And thank you very much for joining us, everyone. Bye-bye.
Thank you so much. Thanks a lot. Bye.
Bye-bye. Thank you.
Bye.
Your certificates within 10 days, inshallah.